Good afternoon. I'm really happy to be here, and Dr. Gibson and all of the, Richard and the whole organization here. So, and it grows every year, and the research grows every year, and it's really getting more and more exciting. So I'm going to be reviewing for you today some of our recent work tying together the gut, the metabolome, and immune disturbances in ME/CFS. And I start with a conceptual framework. So I think about genes, environment, and timing, and how these affect various outcomes, brain outcomes. And through development, we have genetic factors that are affecting the rate of development, but also affect functioning after birth. And I think about environmental exposures writ broadly, a whole host of things: infection, microbiome, xenobiotics or toxins, psychosocial stressors, oxidative stressors, and even your exposure history. What you were exposed to first might protect you or make you more susceptible to something that comes later. And then over time, these factors interact. You have epigenetic regulation as well. Remember, if you were born with all of our genes, but if they were all turned on at the same time, we'd be dead. We're alive because epigenetic turns our genes on and off through methylation and other types of processes. And so the effect here is really outcomes that can happen in childhood, early adulthood, and some of these exposures can be in later life, not just in utero, with disorders in early life like autism, PANDAS, which is strep-related OCD, and in middle life, disorders like multiple sclerosis and ME/CFS, as well as mood disorders, and even late in life, Parkinson's, Alzheimer's, and other neurodegenerative disorders. Why do we think about infection and immune factors in brain disorders? Well, there's a brain-immune interaction. We also know that there have been epidemiologic studies as well as case series that show various brain disorders to be associated with a whole range of viruses, other types of microbes. These have been replicated and shown to be, produce disease-like features in animal models. And there's also the phenomenon of winter-spring birth and also being raised in certain types of environments, as well as nutritionally deprived environments, leading to a variety of other a variety of effects on the brain through an immune axis. So infection and exposure can be bad, but if you don't have any microbes, it's also bad. And so if you're born without microbes in your gut, the microbiome, you have abnormal behavior. These are the germ-free ones, and they're sort of wildly disorganized. And there's a variety of both behavioral as well as biochemical effects, including brain-derived neurotrophic factors, serotonin, ACTH, and cortisol, which are part of your HPA axis. We know that stress response circuitry has been implicated in some studies in ME/CFS, as well as other factors as well. So we focus on certain types of comorbidities in brain disorders that are present in many brain disorders, and it used to be thought that these are just sort of non-specific, but we're really interested in trying to understand the comorbidity, particularly with irritable bowel syndrome, which is up to 90% prevalence for comorbidity in ME/CFS in some studies. But we don't really know, and we need to look at that more carefully. And why would we be interested? Well, there's a gut-brain axis. The molecules that the microbes in your gut are producing include actually little pieces of the bacteria themselves that can sometimes come out of the through the epithelium. There are immune cells and immune molecules like cytokines that are produced. There are even direct electrical connections and wiring through the vagus nerve, which is part of your parasympathetic nervous system, and controlling your autonomic nervous system balance, and as well as bidirectionally molecules that go both ways. And you've seen this many times already. Healthy tight junctions are usually associated with a diverse and more diverse type of bacterial community, and compromised tight junctions are usually happening when you have less of a diversity, and you also may have other types of food products because the bacteria are not taking care of digesting your food appropriately, the wrong types. And this is a really great review by Hansen and Guilleteau, who may be here, in the Biochemist that came out this year. We've been using a whole host of samples. We have quite a large collection now, and we try to get more and more granular on the clinical side as we bring patients in to gather more information. We get clues each time as to what's important. And we learned in some of our early studies that looking longitudinally may be important. Early phase disease versus late phase disease may have different biomarkers, and so we're very keen to do that. We've now, with funds from NIAID, we did collect the samples, but we only had 1 year of support, so we still, we're going to get to, hopefully, to analyze those samples, and a whole host of others as well. One feature that's really important is to think about frequency matching, on key variables. Age, sex, those are the usual ones, ethnicity, race, ones we think of often, but geographic residence and season of sampling. If you're looking at infection, all of those things, there can be clusters of certain types of exposures, toxins as well as infections, and also season can affect those phenomena. For our pathogen discovery and looking at the microbial side, we've taken a sort of staged approach using techniques like VerCap-seq, which is highly sensitive technique, and then if we find something that's new, we'll build a new assay. And we also do bacteriome, or what we typically refer to as the microbiome, the mycobiome, which is the fungi, and then the virome, and using a whole host of starting samples. We started in our work in ME/CFS looking at a range of agents that had been proposed, both viruses and bacteria that had been proposed by the clinician network with whom we were working to be possibly associated with ME. Many of them actually have an association with autoantibody production as well, like parvovirus B19, and also Epstein-Barr virus as well, which is also known as human herpes virus 4, which is the cause of infectious mono. 10 to 15% of people who get infectious mono will not get better and will go on to have an ME/CFS-like course. And we didn't find much. We found some evidence of this, but nothing that really stood out, except in a few small subsets, which we'll be putting together soon in some publications. But we were disappointed to some extent. We didn't find any great new agents either. But we still really think that viruses, as well as other agents, are really key because they signal in the central nervous system, as well as to other receptors, toll-like receptors that are present in the periphery as well, and can have a whole host of effects, and can also lead to autoimmunity, autoantibodies that target the autonomic nervous system receptors, like the beta-adrenergic receptors and so forth. I'm going to focus most of the talk on, on our work on looking at profiles of immune markers. But we also will show some data. I'll also show some data that is, predicted metabolomics based upon our microbiome, our most recent microbiome paper. Another reason to be thinking about immune molecules is that they regulate a host of clinical features that are common either at the beginning of ME/CFS or during relapses, and for some patients remain a problem throughout. Fever production. So these immune molecules are important. They're stimulated by a variety of factors, signaling, usually viruses signal through the Toll-3 receptor, and this viral mimic also does the same. And then there's immune molecules, including interferon gamma, et cetera. And then a certain part of the hypothalamus, where they start hypo-HPA axis, the stress response circuitry starts with your hypothalamus, so it's also the source of stimulation for your immune molecules. And I seem to have broken your laser pointer. Are you going to charge me for that? I'm sorry. Oh, it's still working. Okay. We just got some duct tape. So median preoptic nucleus IL-1 beta, which when I started in immunology back in the Dark Ages. It was before interleukins had numbers. And so we didn't know it as, it was called endogenous pyrogen instead of interleukin-1 beta. So pyrogen is a fire starter controller of the prostaglandin release that controls fever. So even if we don't know the virus, we can look at the downstream signaling molecules that we think should happen if there were a viral infection. And when we can, get some clues as to what might be important. And another factor, another reason is that there are opportunities for these cytokines to get into brain and through the blood-brain barrier and to influence parts of the brain that regulate sympathetic nervous system activity. And that may be involved from a top-down approach, not from the receptors in your vasculature, but a top-down approach that could cause orthostatic tachycardia or dysregulation of blood pressure and pulse. One of our key contributions, I think, in terms of thinking about how you parse out this disease, clearly has heterogeneity in its clinical features, whether it is also due to heterogeneous causes, or is there a gene-environment interaction, or the timing makes a difference. Perhaps it could be the same cause with different genetic factors that alter the expression, or it could be many types of factors. We don't know. We're trying to use our data to help us figure these things out, to use the data that we've collected to parse out the heterogeneity of the disease into what we hope are going to be meaningful subsets. Here, this is older work from 2 years ago, where we found that there was an association with early versus late phase disease. So individuals who either had been ill for 3 years or less, or for longer periods of time, early on, many different immune molecules, pro-inflammatory molecules were increased, and the long duration subjects had this apparent immune exhaustion. So we were, quite interested in that. But it also occurred not just in the pro-inflammatory cytokines, but in Th2, or so-called anti-inflammatory or counter-regulatory cytokines, as well as those that are associated with allergy. And in some cases, there was occasionally, it wasn't just a protein depletion in the long duration subjects, because in eotaxin, which is associated with eosinophil migration and also allergy, that one was increased in the long duration subjects. So they weren't only depleted, but it did look like an overall pattern of immune exhaustion. And we had some signal. These are called odds ratios. Basically, we were predicting the short duration group. So based upon every 1 unit increase in interferon gamma, a virus-associated, fever-related cytokine, there was a 105-fold increased risk of being associated with a short duration ME/CFS group as opposed to the long duration. And there were others as well. We were also interested in how the cytokines played together or didn't play together. And while we found that there was a very dense network both in the long duration subjects and the control subjects, in the short duration subjects, the red lines are the negative associations, the black lines are the gray lines are the positive correlations. And so we only had one negative driver in the short duration subjects, looking like things were dissociated and that they weren't really talking to one another as well as they should. Now that was in the plasma, and we also looked in the cerebral spinal fluid, closer to the brain, closer to where a lot of the action is. And here also, published, it's 2015, actually came out in 2016. But in any case, we found, compared to MS subjects and non-disease controls, spinal fluid of classical subjects with ME/CFS had reduced levels of most cytokines, including almost undetectable interleukin-6, which has a lot of effects actually in memory signaling. So we found that to be really interesting. But again, even some of the molecules like IL-10 that are anti-inflammatory and usually involved in a Th2 response classically, whereas IL-6 can be either pro-inflammatory, anti-inflammatory, even though it's Th2. The IL-10 is decreased as well as the pro-inflammatory cytokines. Okay. And there were differences from the MS groups, although to some degree they resembled one another. This study, we did these samples with Dr. Dan Peterson, who's here somewhere in the back. And what had actually happened when we did the first study is that there was a coding system, P1 and P2, based upon clinical expertise and acumen and 30 some odd years of experience. And the P1 and P2, P1 were the classical subjects that you saw in the last study, and the P2 subjects were these atypical subjects. And these subjects either had an unusual precursor of their ME/CFS, even though they all met classical criteria, this typical criteria for ME/CFS, they had either Gulf War illness or some, a transfusion or a strange encephalitis, or they had cancers in follow-up, atypical MS in follow-up, and other unusual features. And so we separated these groups out and compared them in the spinal fluid. And now these subjects were about the same duration of illness, although there were some that had been less than or equal to 3 years in both groups, so equivalent. And this is the breakdown for the various cancers, immune and infection. And we found here, to our surprise, that we thought you couldn't go much lower, than we had seen in the initial study of spinal fluid. But here we found that the subjects with an atypical presentation or course of ME/CFS had much, much lower levels of pro-inflammatory cytokines that predicted so this was less risk. So higher IL-17A predicted against atypical course. So these subjects had higher levels. Now, okay, we're plumbing now and moving to guess what? Many of you who, as patients, may have participated in microbiome studies, may have seen something like this, but this is how we do it. This is the human microbiome study manner of pulling out the microbiome, and we're interested in understanding how the microbiome and the metabolites that are stimulated by the bacteria directly but into the host indirectly, how the microbiome might drive that. And we know that there's also an important effect in neuroimmune function. All these molecules that are happening based upon these bacterial products, can cross the blood-brain barrier and affect these immune scavenger cells of the brain, the microglia, and also affect neuronal function and serotonin receptors, as well as a whole host of other effects. Why else are we interested in microbiome? Metabolites, what they make, as opposed to what our liver and other metabolic machinery makes. Well, these local effects can create products that affect gastrointestinal motility, cause it to go up or down, reduce your transit time, increase your transit time, and also affect different cell types depending on which layer the bacteria are in. And these are, gases are made by certain types of bacteria versus others, and certain ones will affect, that are tryptamine producers, will lead to serotonin and alter motility that way. So we're really interested in piecing these things together. We also know that there's a possibility that at least a portion of this patient population has an autoimmune basis to their disorder. And so there are different products of the bacteria, butyrate-producing bacteria, that typically are more plentiful in the healthy controls and less plentiful in the type 1 diabetes and other classical autoimmune diseases. And other short-chain fatty acids besides butyrate, propionic acid, acetate, succinate, those drive autoimmunity. So the butyrate is usually protective, and these other ones are not. It's a complicated system. There's many different effects. There's effects on different Th17 cells, which drive inflammation, and a reduction in Treg that may, from dysbiosis, that may alter the protection against the inflammation. And so these have a lot of effects, and the bacterial products can alter oxidative stress and change the environment in the intestine. And you have folate receptors in your intestine, and we think that perhaps those folate receptors are directly changed by the local oxidative stress, and as your immune cells are trafficking through your intestine, that they're seeing these, they're stimulated to create autoantibodies. And certain bacteria, this is just showing, this tryptophan degradation pathway, which I'll show you again in a moment. Some produce serotonin, and then go on to make the circadian rhythm, sleep pattern enhancing melatonin, as well as it's an immunopotentiator as well. But some will drive it down this way through these enzymes, through inflammatory signals, cytokines, glucocorticoids, and other stressors. And these are the types of agents that typically will act through this pathway to drive tryptophan away from serotonin synthesis, away from melatonin. And the balance, though, this is an important effect. The balance is important. You need this system in order to have your memories created and in order to have normal immune function. So it's not an all or none sort of thing. But there are glutamate effects, acetylcholine effects, and a whole host of other neurotransmitter effects that occur. So our most recent study that I'll sort of be closing on before I get to some thoughts about where we're going next is a study on fecal metagenomic profiles. This is done, microbiome studies done in a slightly different way than usual, sort of a high throughput sequencing approach, not targeting 16S regions. And we have a variety of changes, which I'll describe for you in a moment. This is a 50-patient 50 control subset that was nested within our larger chronic fatigue main cohort of 200 patients and 200 controls. So we had a follow-up, and they varied in their body mass index and their presence of irritable bowel syndrome, which was not present in high degree in the controls. And so we had, and the duration of illness was largely, since this was about 2 years after the first study, our 3-year, less than or equal 3-year subjects had graduated to become more than 3 years. We've taken a sort of a hypothesis-free approach, except for we're hypothesizing that there's some fish to get here, but we're using an analytical tool called IASD that uses sort of the shape of data to try to help us find subsets that might exist between the biomarkers and certain subsets of ME/CFS that we hope will have some clinical meaning. And we did find, using this technique, Dora, one of the postdocs in our lab, Dora Negisagal, had found that there was an association with, different associations with ME, with irritable bowel syndrome versus those without, and the controls clustered generally very tightly together. And we found when we looked at these groupings, this is ME/CFS versus controls. We found there's a lot of bacteria here, of course, but we found some things that had been found before, that there were increases in allostipes that had been found in, we found that in all of the groups, and whether they had IBS or not, or whether it was all together. And we also found some reductions in fecal bacterium, replicating some work in other groups, including Maureen Hansen and Ludo Gilitas.
Five, Mady.
Okay. And we also found there that there was vitamin B, the predicted bacterial metabolites were driving vitamin B6, which might mean that there was an overdrive in vitamin B6 dependent signaling. And for some reason they were using it up and needing a lot of it. But it's also an increase in this atrazine degradation. And atrazine is a pesticide. We heard about glyphosate earlier, Roundup, and so there's a lot of pesticides in our food chain, and so we're very interested in trying to understand whether there may be cofactors or that these could potentially serve as a driving factor. And just in conclusion, we had different factors and different symptom profiles that clustered together in the various groups with and without IBS and without BMI. So we're really interested in trying to use this to follow up and also look at some irritable bowel syndrome patients who do not have ME/CFS to do that. And we're now have plasma metabolomics that will reflect the actual quantitation, if you will, or at least relative quantitation of the host and bacterial metabolites in the blood through a collaboration with Oliver Fien. These metabolomics, look through a whole host of different factors, and I just wanted to, end really thinking about where we're going with this. We're mining our entire database, all of the biomarkers, trying to gather all of the clinical information that we can. We're trying to add in newer studies and follow-ups in the same subjects, to add in, using digital sensors, mobile or web tools that allow bedbound patients, or even just people who don't, just to give more information that goes along with their symptom rating, so we can understand the course of illness and how that relates to the different biomarkers. And trying to derive predictive biomarkers that tell us something about perhaps treatment response, course of illness, as well as other factors. And so we're using a wide range in a systems biology type of approach. And one of the studies that we're doing is really what we call sort of a laboratory in a blood collection tube, is designing studies. This is a little test tube, if you will, but it collects blood directly. Here's the needle. And then this blood, once the blood is collected, it goes in a little heating block, just like it would be a little incubator. And you can put stimuli in there. You could put toxins in. You could put the pesticide. You could put atrazine in there. You could put viruses and different bacteria or bacterial products, and then look at the response. And we do, again, this systems biology approach, both genetics, epigenetics, as well as gene expression. And we're really keen to try to piece together what are the vulnerability factors, what are the exposures that have been most likely, and can we add in toxicology as well to look for in the metabolomics. You can usually get toxicologic signatures as well. And again, moving to the gene-environment timing interaction, right? We're thinking about how these factors interplay across a lifetime to yield a disorder such as this. And this just came out in JAMA, about precision medicine. And they're always focusing on these rare variants. And they may be important, but when you're looking for these gene-environment interactions, it could be a common exposure. A lot of people get infectious mono. This happens to be on MS, but this is where we want to go ultimately. But this is MS risk from environmental factors and the association with HLA genes, a certain type of gene. Could be an uncommon exposure with common gene variants. And is timing important in terms of the interaction of the gene and the environment interaction? And then if the gene variants or the exposures are undefined in your study, we have to recognize that our power is very reduced. You usually need 400% larger study populations in order to be able to detect a moderately strong gene-environment interaction. And so we have to keep that in mind, having large numbers, collaborating, and trying to come to using our good sense in terms of leveraging the resources, including the patient samples. And this is my final slide, which is really, again, in MS, but this is where we need to go in ME/CFS. We need to take the gene-environment interaction. We need to take our environmental exposures, understand how these affect immune cells, cells of the epithelium, how that leads to pathology in brain and other tissues, what autoantibodies are created, and put that together with what we know about CNS inflammation. And I love closing on this quote. It's a great quote. And, so Einstein students said, questions are the same as last year. How can you do this? True, Einstein said. But this year, all the answers are different. We must keep an open mind. We can't just say, oh, they did that 20 years ago. It was, there was nothing to it. Our technologies are getting more sensitive. We're more aware of the controls that we need, and also more aware of all the intellectual resources as well as the funding that we need to cross the globe to make this a success. Thanks very much.
Okay, there's time for questions.
Okay, you collected the samples. how are you getting money?
Well, you know, our group and other groups have applied for center grants and proposing to use some of those samples there. We're also applying for other NIH funds, but, you know, we're also looking to private donors for, you know, for help, and it's a really critical time right now, and it's really clear. We also have, through the Microbe Discovery Project, we've really had, you know, some critical support there that has, through crowdfunding, that has been really essential, even in just helping us to collect these samples, bank them, and characterize the subjects, and to have them provide information to us as well over, you know, 4 time points over a year. But, you know, we're trying to get creative, and I think that we need, I think, to, you know, enhance the research community and to partner together as much as we can, and to, you know, I think that I really do think that there's, you know, it's starting to change, you know, and I think that as long as we can get through the critical periods and start to convince other disease, you know, other groups that are focused on, let's say, infectious diseases broadly or neuroimmune interactions more broadly, that this is an important disease that can help us in understanding a host of other diseases, as well as answering a vital question that, you know, for this disorder that disables so many people.
Okay, thank you very much.
Okay, thanks.